All right, guys, let's get ready to science. Yeah, biology is not out. All right, guys, before we get started on what we're going to do today, let's review the last lesson. All right, so we're talking about cellular energy. So we're starting off with, of course, an introduction to metabolism. So what is metabolism?
The overall
the overall rate of, or no.
Well, that sounds kind of COVID-ish.
Oh my God!
That's what I said this morning.
cover your nose.
That's what I said this morning. That's what I said this morning.
I will come cough on you.
Well,
Alright.
and it won't affect them if you don't cover your nose.
All right, guys, let's settle down. All right, let's go back. What is metabolism? Anybody?
Hold on, make sure.
I don't want you to look it
The
up. Be
overall
real.
rate of the cells dying and regaining.
Nah. At least
It's the
try it.
overall
Come on.
rate of something.
Is it a rate?
I thought it was.
Of the
Metabolism is the totality
of energy.
of the cell's chemical reactions. It's all of the chemical reactions. And so that's all of the breaking down and all of the building up. That's what metabolism is, okay? So we're talking about metabolism, so you have to keep that in mind. When we're talking about metabolism, we're looking at how things are breaking down, well, actually what's happening to the energy as things are being broken down or built up. All right, now, what are the two types of metabolism?
Catabolic and anabolic.
All right, so I have anabolism and catabolism. All right, so what's the difference? In an anabolic reaction, what's happening?
Building up.
Building up. All right, so if I have an anabolic reaction, so we know that cata breaks and ana builds.
Yes.
All right. Now, what type of chemical reaction is going to be happening during an anabolic reaction? Is it going to be a dehydration synthesis or a hydrolysis reaction?
Dehydration.
Dehydration. So we're going to have a dehydration synthesis. All right. And tell me what we know about anabolic reactions with relation to energy.
They take energy. They store energy.
They take energy, they store energy, so they are energy consumers. So are they endergonic or exergonic?
Endergonic.
They're endergonic. All right, so they are endergonic. So we know because they are energy consumers, they take in energy, that the amount of energy available in the products at the end of the reaction is going to be higher than the amount of energy that is available in the reactants. So since we're talking about energy, what is energy in the first place? What is energy?
The ability to work.
The ability to do work or cause change. And what's the ultimate source of all energy in this ecosystem and on Earth?
The sun.
The sun. All right, so energy flow is one directional. Energy enters into the ecosystem in the form of light energy, and it exits the ecosystem in the form of what?
Heat.
Heat. All right, so it's one directional. Now, can we make energy?
No.
No. All right, so there's laws of thermodynamics, and according to the first law of thermodynamics, energy can neither be created nor destroyed. It is only transformed or transferred, okay? According to the second law of thermodynamics,
Entropy.
even if things become more ordered as they become more complex, the universe itself becomes more disordered. So with every energy transformation or transfer, there's an increase in entropy. What is entropy?
The disorder of the system.
Disorder of the system. So why? How? As my system, as me as a system, as I became more ordered, the universe became more disordered. Why? What happened?
Heat.
Heat. What does heat do?
Makes
Oh, it speeds up.
Makes stuff move around.
Makes stuff move more randomly. It speeds up things. All right. So again, heat is a type of energy. So What are the two main types of energy?
Oh, kinetic and potential.
Kinetic and potential. What is kinetic energy?
The
Movement.
yeah, movement.
Energy of motion, right? So when molecules are in motion, now the faster molecules move, the more randomly they actually bump into each other. And we know that an increase in heat causes the molecules to move faster. So with every energy transformation and transfer, there's a release of heat to the environment. So the more heat are released, the more random things will move, okay? So that's kinetic energy. But thermal energy is a type of kinetic energy. What about potential energy?
Energy that's yet to be, or it's like at the top, like
Stored.
So potential energy is the energy based off of position or structure. Okay? Position or structure. Potential energy is stored energy, right? So we can transform from one type to the next. Question?
so like if there's a really electronegative compound, it has a lot of potential energy.
Right. Right. Alright, so chemical energy is a type of potential energy. Okay, so energy enters the atmosphere, enters the ecosystem in the form of light energy, but we are not able to actually use that light energy. What types of organisms are able to capture and transform that type of energy? Autotrophs. All right. Auto, self, troph, nourish. They are able to transform light energy into chemical energy stored in the bonds of the molecules that they create. And then organelles such as the mitochondria are able to transfer that chemical energy from the bonds of things like sugars into the bonds of the cell's energy molecule, which is adenosine triphosphate, or ATP. Now, we talked about anabolic reactions. We know that anabolic reactions require energy to be input. They're endergonic. So that means that there is a change in the amount of energy from the products, from the reactants to the products. That change in the amount of energy is called free energy. Free energy is the energy that is available to do work. But that's not the only type of energy associated with a metabolic reaction. What type of energy is required to get a reaction going in the first place?
Activation.
Activation energy, that energy of activation, or E sub A, the energy of activation. Now, there are molecules called enzymes that act as catalysts. What does that mean? What do they do?
Lower
They
the
speed up
activation
the-
energy.
They speed up the chemical reaction by doing what?
Lowering the activation energy.
Lowering the activation energy. Enzymes make life possible. Okay? Activation energy has to be absorbed from the environment to get a reaction going. Without enzymes, it would take too much energy to get the reaction going. It would take a lot of heat, so you would get too hot. It would take a lot of energy to get the reaction going, and so that's going to increase the amount of time that it takes for the reaction to get going, because it has to sit there and gather all the energy it needs to get going in the first place. The way an enzyme works is it lowers the amount of activation energy required to get the reaction going. Now, enzymes act on the energy of activation, which is from here to here if it's uncatalyzed, or here to here if it's catalyzed. Enzymes do not interfere with the change in free energy. The amount of free energy that is available will be the same from the beginning to the end of the reaction as it would have been with or without an enzyme. Do y'all understand what I'm saying? Enzymes do not change free energy. Now, I've drawn a particular type of reaction. Would this reaction be anabolic or catabolic?
Catabolic.
Catabolic, how do you know?
It goes down.
What goes down?
Free energy.
The free energy. The free energy change. The change in free energy is negative, right? Or it's less than zero. So it went down. So that means the energy that was available in the reactants was more than what I had left in the products. So that means the energy was released. That makes this an exergonic reaction. Now back to anabolic, exergonic catabolic reaction. Back to an anabolic reaction.
Can you draw what, you said that one was catabolic, what would the...
I'm about to do with both of them right now, little fuck. You read my mind.
Best friend.
You go up, Ryan.
Ryan, just like that.
Right there.
All right, uncatalyzed, catalyzed. So, energy of activation, energy to get it going, then it continues to absorb energy. So the amount of energy continues to go up. So I have my E sub A here, right? Took less to get going, still goes up. And this change from here to here, this is my free energy of my reactants, and this is the energy of the products. The reactants have less energy than the products, so the amount of available energy increased as the reaction continued. Do you see that? Yes.
Why is the R right there and not on the bottom?
For reactants, I just wrote it right there because that's, yeah, I mean, just to show that reactants have less than, yeah. All right, now, so we know that energy is absorbed. So we know that this is endergonic, and we see these type of reactions when the reaction is anabolic. So would this happen spontaneously or non-spontaneously? Non-spontaneous. Anytime delta G is positive or greater than 0, then we know that this is a non-spontaneous reaction. Okay? So those are words you need to associate with each other, right? If it was a hydrolysis reaction, that means it was building something up. If it's building something up, that means it's anabolic. If it's anabolic, it has to absorb energy, so that means it's endergonic. And so that also means that it is going to have a change in free energy that is positive. So if the change in free energy is positive or greater than 0, that means that this reaction is non-spontaneous. Now see how all that goes together. All right? So in this case, energy is being absorbed. Now what about the other side of the coin? All right, so in this case, you have catalyzed, uncatalyzed, right? So what happened to the amount of free energy?
It went down.
It went down, so delta G is negative or
Less than 0.
like greater than, less than 0. I have to do my greater than sign sometime. Greater than. Boy, I'm sorry, anyway. Like a lot of times, just if I'm doing greater than, less than, I always throw the greater than. That lets me know the other one go the other way. Because, you know, my right hand is greater than my left because I'm a righty. That won't work if you're left-handed. If you're left-handed, it don't work. Yeah, that's how I remember it. My right hand is greater than. All right, anyway, moving on. So, what's happening to the amount of free energy?
Going down.
So, delta G is less than 0. So, is the energy being absorbed or released?
Release.
So, there's an energy release. Do I expect this to be kata or ana? Kata, because when kata breaks, when bonds break, energy is released. Right? Catabolic reactions break bonds. When bonds are broken, energy is released. So it's catabolic, energy is released, spontaneous or non-spontaneous? Spontaneous. What else can you tell me about it? So is it exergonic or endergonic? Exergonic. Did we cover all of the bases on this one?
Yeah.
All right, so now, give me an example of a process that is catabolic.
Cellular respiration.
Cellular respiration. Give me an example of a process that is anabolic.
Photosynthesis.
Photosynthesis. All right, so do you see how metabolism relates to those two processes and why they tie in together with your project? All right, so been down. All right. So anyway, now we talked about enzymes, right? So enzymes actually lower the activation energy and shorten the time that it takes for a reaction to happen to make life sustainable. Okay, if it was not for enzymes, it would take too much heat, too long for processes to happen, so life would not be sustainable. But what is an enzyme? We talked about 4 major organic compounds in a previous unit. What were those 4 major organic compounds?
Macromolecules.
They are micromolecules, but what are they specifically?
Proteins, lipids,
Nucleic
nucleic
acids.
acid, carbohydrates.
Right, carbs, lipids, proteins, nucleic acids. All right, enzymes are a special type of one of the 4. So which one is it? It's a special type of protein. And we know that proteins are sensitive, right? They do all the work, but they're really sensitive about, they are specific about where they do, the conditions they do their work in. All right, so when we think about enzymes, because they are proteins, they're just as specific. Now, there's 2 levels that they won't work in, but they won't just have a complete melt breakdown. You know, you ever seen somebody that, like, some people just sit there and just won't do it if the stuff isn't right, but then other people have a complete just meltdown. All right, depending on how you treat an enzyme, it's going to do one of the 2. Now, the first thing, it is specific for the structure of the substrate. That's called lock and key, okay? It's a lock and key mechanism. Now, what an enzyme does is it has, where does a substrate bind to an enzyme?
At the active site.
At the active site. So when the substrate binds to the enzyme at the active site, the enzyme will actually make a conformational change in shape to make that what's called an induced fit, to make it fit perfectly, okay? But it only does that if the right enzyme binds to the active site. It's like a lock and key mechanism. So they are specific for the structure of the substrate. If the wrong thing tries to bind there, it won't do anything. The next thing they're specific for is the concentration of substrate to enzyme. Now, it doesn't mean that they won't work because, you know, there's substrate there, but the reaction will start to slow. Like, once there's less and less substrate available, the enzymes work slower. Like, there's nothing for them to, you know, like some of them sitting around doing nothing while some of them doing the work. Kind of like y'all gonna find with y'all group project. But hopefully not, this is an AP class. Y'all should be good. But, so how can you overcome, how can I speed up this reaction? What would I need to do?
Add more substrate.
Add more substrate, right? So, but what happens if I put too much substrate and all the enzymes are bound with it? Like, that's not good either, right? So there is a concentration of enzyme and substrate that the enzymes prefer. Now, these other ones will cause it to have a complete breakdown, all right? First thing being pH. What does pH stand for? The percent hydrogen ions present in the solution, right? All right, so what's the scale? 1 to 14. All right, and so 0 to 14. So we have acidic and alkaline, right? So you can have one side is acidic and the other side is alkaline or basic. All right, so what's the pH? What's the range for acidic?
0 to 7. 0 to 6. Or 6.9.
Yeah, something like that. What about alkaline?
7.1 to 14.
Right. So we know, and 7 is neutral, right? So we know how the pH scale works. Now, enzymes are brats, right? They gotta have the perfect conditions. So if you have an enzyme that is used to being in something alkaline or whatever, you may see something like this. So let's say 7, 8, 9. You may see it on a graph, right? Now, they have a range that they will work within, but once they get outside of that comfort zone, they denature. What does denature mean? They lose their structure. They unfold, right? Bonds begin to break. They let go. Like it's complete mental breakdown. All right, but some enzymes are able to renature. What does that mean?
They will kind of get better.
Refold. Yeah, and when the conditions are right, some can actually renature. Not all. Some things break down and that's is damaged. Now, what you can see on a graph though you can see what their preference is. Whenever you're looking at a graph, the highest point on the graph is called the optimum conditions. So if we're looking at pH, that means that enzyme is going to work best at about a pH of 8. Doesn't mean it won't work at 7 and all the stuff in between. Doesn't mean it won't work at 9 and the stuff in between. What that means is if you want it to work best, it works best at 8. When you get outside of this range, it will begin to denature and it will not work. They have a range for pH. There's also a range for temp. And there's a range for salinity. Do we know what salinity means? The concentration of salt. Now, let's discuss the temp thing, okay? Let's relate it to something that I'm pretty sure we've all had to deal with before. Fever. What is fever a sign of?
Your body's working against the infection.
Your body is working. So it's a sign that your immune system is working against something foreign inside of your body. Why do you think you have a temperature? Let's think about energy and what we know about the laws of thermodynamics. What's happening?
Are your cells moving faster to try to counter?
Well, the cells, which cells? You mean like the white blood cells and the T cells? The cells of the immune system, they're doing more than they normally do. So they're transforming more energy than they normally transform. And what happens every time energy is transformed and transferred? Some is lost as heat. So what happens when that heat accumulates on the inside of the body is the core temperature is raised. So it's not that the fever is a bad thing, because the fever is a sign that your immune system is doing what it's supposed to do. Here's the problem. These little temperamental guys right here, they don't like it when you change their core temp. They don't like it when you get outside of their range. So it's not that the fever was bad. A low-grade fever is cool because it keeps within the range. It's a high fever or a fever for an extended amount of time. That's the problem. So these guys begin to unfold as the core temperature begins to rise, right? Now there's different enzymes that have different thresholds, so different things will denature first. But if they begin to unfold, guess what they stop doing?
Functioning.
They stop functioning. That can lead to seizures and organ failure and all other kind of crazy stuff, because this little temperamental mug right here didn't want to let the immune system do what it was supposed to do for the time it needs to do it for. Okay? So, do you see how even what you're learning in the energy there's energy transfers that's happening because there's more work being done. So that's going to increase the amount of entropy and the amount of heat that is being released that your body normally wouldn't do. The goal is to maintain homeostasis, keep everything within that range. But if something goes bad, then it has to do something to try to get rid of that thing so it then gets stuff back into the correct range. All right, why would salinity how could salinity be an issue?
Because the tonicity of the cell.
Because of the tonicity of the cell. Remember this, if you don't remember nothing else, water follows salt. Okay? So as your body attempts to remove excess salt by sending it out through the intestine, guess what's going to go behind it?
Water.
The water. Diarrhea. My daughter used to watch something like that. I don't know what it was. It's like, why are you watching? He's standing in the shower and something, diarrhea. I don't remember what it was. I forgot I recorded. Whoop, whoop. And I don't even know how to edit, so that's going to be in there. All right, so anyway, what was I saying? We were talking about enzymes and their temperament. All right, so they're temperamental. They really need to work within the correct range. That's a good thing, though, because you don't want enzymes that function in digestion in the stomach to function for digestion in your bloodstream. Like, there's two totally different pHs, right? So you don't need those digestive enzymes that function in an acidic environment to get into your blood and then just start breaking down everything around it, because it's like, hey, party over here, free stuff, woo, red blood cells, white blood cells, macrophages, yay. All right, anyway, so now enzymes we know are a class of proteins, so that means they're composed of Amino acids, right? Now, what do we know about amino acids? They have these things called functional groups, and they have R groups. Now, the R groups are actually what the interactions between the R groups are what actually makes the enzymes fold into their conformations. When enzymes bind to substrate, they form an enzyme substrate complex. And we said that an enzyme was a catalyst. So, what is a catalyst again?
Something that lowers the activation and speeds up the reaction.
How?
Lower the activation.
Okay, how?
By breaking apart substrates.
That's one way. It doesn't actually break it apart, but it does strain the bonds in the substrate. All right, it does strain the bonds. So this will be in what type of reaction? Catabolic or anabolic? Catabolic. All right, so it can't strain the bonds in an anabolic reaction because you're trying to make bonds. So that was one way. All right, what's the other one? There's actually 4 ways. I call it COPS.
Forming the bonds.
Oh, you
So first thing is it covalently bonds to the substrate, right? It covalently bonds to the substrate. That's in the active site. All right. What's another way?
open.
Not open.
I was just saying.
It orients the.
Oh God, substrate scramble.
Is that the right word?
Orientate.
Orient.
That's not.
That ends in I.
Oh, you didn't spell that.
Is that right?
Orientes. It's
No,
orientes,
that's
no
right.
E.
Yeah, there's no E.
Yes.
Okay.
Correct. On the Orient Express. And then
All right. What do we mean? All right. So let's think about our friends that participate in a 1,4-alpha glycosidic linkage. 1,4-alpha glycosidic linkage. Which one of the 4 major organic compounds makes glycosidic linkages?
Lipid.
it's carbohydrates.
Carbohydrates. Lipids make ester linkages. All right. So the 1,4-alpha bond is found in glucose, between the alpha glucose. That's the bonds that we can break down, right? That's the bond that we find in glycogen. The bond. So 1,4 tells me which carbons the bond has to be between. So that means one of the glucose molecules is going to use its number 1 carbon, and the other glucose molecule is going to use its number 4 carbon. So that means in order for that bond to form, one molecule must be turned so that the number 4 carbon is facing the number 1 carbon. They have to be oriented in the correct position. Now, what an enzyme can do, since it has an active site, is create the active site so that when one of the glucose molecules bonds, the number 1 carbon has to be this way. And when the other one bonds, it has to put its number 4 carbon that way. So that when they actually bond to the active site, they're already turned in the correct way. That's going to make that bond easier to form. Because if this was a 1 carbon on this side and then right next to it was another 1 carbon, the other somebody got to turn around so we can actually make this bond, right? More time to figure out which way we're supposed to be going. So by orienting the substrates correctly, it eliminates the time that it takes to do that. Okay? So, would this happen in an anabolic or a catabolic reaction?
anabolic.
Anabolic, right? So we know this is going to happen regardless in both. This one happens in anabolic. This one happens in catabolic. One more thing. What does it do?
Provides the right environment.
It provides the correct microenvironment. That's right. So when it actually makes that conformational change in shape to induce the fit of the substrate, the environment that's inside of that active site, of that substrate the enzyme substrate complex can be slightly different than the environment that's outside of the enzyme. So it provides the appropriate microenvironment, okay? My hair on my nose, itch. Alright, so those are the 4 ways, okay? It covalently bonds to the substrate, orients the substrates correctly, provides a favorable microenvironment, or it strains the bonds in the substrate. This one, the P and the C, is going to do each reaction. O and S depends on if it is anabolic or catabolic. Make sure you know those 4. Alright? Alright, now, enzymes. Do we want it always doing this? Do we always want our enzymes breaking down? Assisting, because they don't actually break down. Let me not say it that way. Do we always want them assisting with the breakdown or buildup of things? Always. Non-stop. So, in order to control that, they have to be regulated. Okay? Now, enzymes can be assisted, though. They can be assisted, or they can be turned off. Alright? Anything that turns off an enzyme is called an inhibitor.
Competitive and non.
Anything that assists an enzyme is called a cofactor. So you have inhibitors and you have cofactors. Let's talk about cofactors first, even though I wrote it over here. A cofactor is something that helps the enzyme, okay? Cofactors could be organic or they can be inorganic. What's the difference?
Organic has a carbon.
Organic has carbon. All right, if it's an inorganic cofactor, we call it a cofactor. That's it. If it is organic, we call it a coenzyme. Coenzymes are just organic cofactors. So if it's inorganic, like vitamin A, vitamin K, vitamin B, those are enzyme helpers, all right? So if it's inorganic, we just say they're cofactors. But if it is something that is organic, we call it a coenzyme. Wait, the vitamins are coenzymes. The cofactor should have been like ions. All right, so yeah. No carbon, cofactor. Carbon, coenzyme. Yes.
Think of the bathroom.
When I finish this, best friend. All right, we almost done.
Okay.
Inhibitors. There's 2 types. What are they?
Competitive and non-competitive.
Competitive and non-competitive. Competitive and non-competitive. Now, both of these,
You forgot it.
No competitive. Erase the whole erase part.
I don't know what y'all talking about.
Non.
That said non-competitive. It don't say no competitive. It said none.
Let's
Y'all
check the
can't see.
replay.
No, that's not. Well, that's not. We outlaw and replay. It makes the game last longer. All right. Anyway, so competitive or non-competitive. All right. Now they both have to do with structure. Okay. In a competitive inhibitor, the inhibitor has a structure that is similar to the actual substrate. So it actually competes with the substrate for the active site. Remember, the active site is for something that has a particular structure. So the structure of the inhibitor is going to be similar, very similar to that of the substrate, and it competes with it for the active site. And if it's bounded to the active site, then the substrate cannot. Now, you can overcome competitive inhibition by increasing the amount of substrate, because that increases the chances that the substrate will get to the active site first. You can have things like poisons, toxins, and stuff like that can actually act as competitive inhibitors. There's this one, I have to look it up. I meant to look it up last block, but I forgot. But there's this disorder that you can actually die. It's a bacteria that actually you get infected and it causes like severe diarrhea, right? And the way that it works is it actually inhibits the inhibitor